How's it going, everyone? And in this video we are going to be walking through the overview of cytology. And cytology is a generic name referring to the structure of human cells. So to get a little bit more specific here, what we're going to be looking at and taking a deep dive into are membranes, the cytoplasmic matrix, which is also known as the cytoplasm. We're going to talk about the nucleus, the cytoskeleton, mitochondria, ribosomes, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi Apparatus, as well as vesicles. And so I have my quiz notes here that you guys can refer back to, but for the sake of kind of walking through and talking about this stuff, I think it's best to go through a little Anki deck that I created on this. Guys can download it for free and review this on your own time. And I think Anki is a great way to get this material learned. So basically, I'm going to start off with membranes and what are membranes. And so basically, membranes are these things that are referred to as amphipathic. So like an amphibian is able to be on both land and water. Amphipathic means that they are both polar and non-polar, and specifically membranes have these subunits called these phospholipids that have a polar head and a non-polar tail, and then they come together and they form a phospholipid bilayer, which separates the cell from its outside world and gives you a cytoplasm, that inner volume of space inside of a cell. And then membranes are also used to form vesicles for moving proteins around and creating certain types of environments inside the cell so that they can do certain functions. So that phospholipid bilayer is a very important thing to make note of when you're discussing membranes. And so the next thing to look at are, we've got certain types of proteins you can embed into the membrane. And so inside the cell, you can put like a G-coupled protein receptor, for instance, or you can put some kind of ion channel to allow ions into your cell. So you can put a lot of interesting things into here, and that is part of what makes membranes interesting and what allows us to live with our cells. So these proteins can get embedded, and then you have different types of proteins inside of your membrane. So you have things that are referred to as integral membranes, and these are things that go through both layers of that phospholipid bilayer. And then you've got things called peripheral proteins that are those things that are just on the surface of that phospholipid bilayer alone. So that is a key thing to make note of when you're learning and looking at membranes. And so another thing that is interesting to talk about in membranes is cholesterol, because cholesterol, as we've probably heard from dietitians, is something that you usually want to keep low, but not too low, because your membranes actually do need it. And so cholesterol is this thing that affects the fluidity, the thickness, the compressibility, and the water penetration of the phospholipid bilayer of your membrane. And so basically, as you increase the amount of cholesterol, it changes the characteristics of these membranes in such ways that it can sometimes be detrimental to your health, which is the reason why you need to be careful with not eating too many red meats, for instance. And so if we were to take a look at the inside of the cell, the cytoplasm as we call it, is a bunch of water, enzymes, macromolecules, ions, and we've got some dissolved gases like carbon dioxide and oxygen, and we call this the cytoplasmic matrix, which I think a lot of people just call the cytoplasm, but some people call it the cytoplasmic matrix. And so when we take a look at our cell and we look at one under a microscope, sometimes we're going to see this darker region, and that's what we're going to call the nucleus. And so inside of this nucleus, we've got DNA that we're storing. DNA is this genetic material that encodes for all these proteins that help you survive. And so this is the nucleus. This is what it looks like if you were to take a look at a microscope right now. We call it like 100x. And so that's what you see. The other interesting thing that's happening inside of this nucleus is that you're getting your DNA transcribed into RNA, which is going to leave the nucleus, go into that cytoplasmic matrix, and then be translated into a protein. And so when we look at the nucleus, we're going to see that there are non-randomized distributions of DNA, or collections of DNA, that are only visible during mitosis. And so these are referred to as your chromosomes. So typically the DNA is so unwound that you can't see it because it's super, super skinny. But when you begin to enter mitosis, when your cells are about to divide, this is when your DNA will begin to coil up around histones and form chromatin. And this chromatin is this darker thing that we can all see under our microscopes. And that's what we call our chromosomes. And the other thing that's interesting about this is if you were to look at the area inside of the nucleus, you're going to note that a lot of these chromosomes, the DNA itself, is not randomly scattered. It's segregated into these specific territories that people refer to as chromosomal territories. Now the next thing is the nucleolus, and inside that nucleus there's an even darker subregion of it, and inside of this nucleolus is where we make ribosomal or RNA, and this stuff eventually leaves the nucleus to become these ribosomes that actually manufacture the proteins that take that mRNA from the DNA in the nucleus and then translate it. And so this is kind of a graphic representation of what does it look like when we have your DNA and how it coils around these things called histones, and we get two types of chromatin. So we have a really dense form of chromatin, and we call that the heterochromatin, and then we have the looser packed DNA, or less tightly packed DNA, and we call this euchromatin. And the Greek prefix eu means that it's true chromatin, and I like to think of this, helps my brain think through it, is that it's basically the true chromatin, and what that really means is that these are the genes or the DNA that truly gets expressed, because as you can see in this picture, the DNA is so loosely packed that it is physically or spatially possible for ribosomal or RNA polymerase to actually latch onto this thing and transcribe it into mRNA that could actually make a protein. So when the DNA is all tightly packed up like that, you don't get a chance for RNA polymerase to actually get in and do anything because there's just no room for it to work. But when you unravel that DNA and you turn it into euchromatin, the true chromatin, that's the stuff that you can actually translate into proteins ultimately. And so, as we were just saying, you've got two different types of chromatin. We've got the euchromatin that is easily reached, and then we've got the heterochromatin that is densely packed and won't ever get expressed because it's just too packed up way in there. And then when we zoom in on our nucleus, we're going to make note of a double lipid bilayer membrane, and this is going to be referred to as your nuclear envelope. And so your nuclear envelope is this thing that differentiates your nucleus from everything else inside that cell. So it's the barrier between the cytoplasm and the actual DNA and the nucleolus. And so this is a very important structure. It's regulating what goes in and out of that nucleus. So when we look at viruses, for instance, somehow these viral genetic payloads need to make their way from the outside of the cell through the membrane, and then from the membrane in that cytoplasm, they need to figure out how to break through and get into your actual nucleus. So they have to get their way past an additional barrier, which is that nuclear envelope. And so that's an important thing to make note of. The other thing to say here is that with your nuclear envelope, this little membrane you've got on the inside of your cells, when you're doing mitosis, this whole envelope disintegrates because you want to move the copied DNA, or half of the copied DNA, to one half of the cell and the other half of the copied DNA to the other half of the cell. And so you can't have that wall still in the way. And so what happens in these cases is that you phosphorylate the actual lamins, these things that are inside of the nuclear envelope, and this breaks them down so that the inner volume of the nucleus is now accessible to other things. And so the other key thing to make note of here is that inside of these nuclear envelopes, we've got nuclear pores, which are really, really tiny, and they have tons of proteins inside of them to regulate what goes in and out. And the other thing to say here is that water and ions are able to readily diffuse through this thing, so you don't need to burn any calories to make that process happen. However, when you want to do larger molecules like proteins and RNA, these are things that you need to actively move out of the nucleus or into the nucleus, so it takes more energy and you need proteins and you're burning calories to make that happen, so it's called active transport. Now, we've got three types of cytoskeleton filaments, and so before I say this, like the main purpose of the cytoskeleton is to basically give your cell structure, to make it have support so it doesn't just collapse, so it's like your own skeleton, so you're not just a blob. And then it's also supposed to add tensile strength, so you can't just pull this thing apart. It's supposed to allow your cells to be able to move to new places if they need to, and stuff like that. So we've got three types of cytoskeleton filaments. The first is going to be called actin, and actin are very thin filaments that are, one of their roles is to be involved with your muscle cells, the actin myosin form cross bridges that actually contract. The actin will contract on those myosin fragments and allow you to flex your muscles or extend your muscles. And then we've also got microtubules and intermediate filaments that we will also discuss later. And so when we take a deep dive into actin, we're going to see that it has these subunits that we call G-actin. And G-actin monomers are going to come together and they're going to form their trimers. And so with these trimers, you're going to ultimately form these double-stranded helical fragments that cross-link to form things that are called F-actin. And F-actin, which generically is just called actin because at this point it's considered the full structure, has a polarity to it. And so what polarity refers to is a difference between the chemical properties of both sides of the molecule. So if this was like a perfectly symmetric molecule, if they had both identical properties, it wouldn't be polarity. But in this case, actin is one example of a filament that does have polarity. So we make note of that when we're analyzing the structure of this. Now, organelle movement, chromosome movement, which is used for mitosis and meiosis, or cilia and flagella structure and movement, comes from this thing called microtubules. And so these microtubules are pretty interesting because they are kind of like these rods, and they can, really provide that strength and support for the cells. And inside of these, or on these actual microtubules, you can have proteins that will move stuff. These motor proteins will move things back and forth. And so you have this thing called dynin, and dynin will move to the negative end of the microtubule because microtubule, just like actin, is considered a polar molecule, or polar filament, meaning it has chemically different ends to it. And so as these dynin motor proteins carry things, they go to the negative end of it, which is to say they're going near the center of the cell or the inside of that cell. And then you have kinesin motor proteins that are going to be responsible for moving stuff towards the plus end, or positive end of these microtubules, and that is considered to be outside of the cell. So if you wanted to move away from the center of the cell, you're going to take a kinesin motor protein, and a dynin motor protein would take you closer to the inside of the cell. And so when we take a deep dive into microtubules, we're going to note that they have two subunits that are referred to as alpha tubulin or beta tubulin. And so these two things come together to form what's called a heterodimer because they're different. They're not just the same monomer. And these heterodimers are going to arrange into these filaments, protofilaments, so early filaments that are arranged into cylindrical structures that we eventually and ultimately will call microtubules. And so we're just coming back here to review a little bit on what membranes are so we can beat this into our memory. Membranes are amphiphatic, meaning they've got that polar head, the nonpolar tail, and they're forming a different environment for cells. They're forming that phospholipid bilayer for their cells. And so we've also got our integral proteins, which are spanning the entire membrane, whereas we've got our peripheral proteins that are just those things that are anchored on the surface. And then we have, getting back to microtubules now, we have cilia and flagella that consist of 9 pairs of microtubules with 2 microtubules in the center. So you can see in this picture right here how you've got 9 pairs around the outside, and then on the inside you've got 2 central microtubules. And so these tubules, the pair, you will call one of them A tubule and the other one you'll call B tubule. And what differentiates these two is that the A tubules have 13 protofilaments, whereas the B tubule has 10 to 11 protofilaments. And when we look at this overall 9 by 2 plus 2 structures, the way I call it, the 9 pairs with the 2 central microtubules, this is called an axoneme. And so again, talking about cholesterol, we have cholesterol, which affects the fluidity, thickness, compressibility, and water penetration of the lipid bilayers of our membrane. And then blank, we embed proteins into our cell to help with transport, anchoring, and cell signal reception. And then when we look at centrioles, so centrioles are a little bit different from the microtubules that we were looking at earlier. And the thing with these is that they have 9 triplets of microtubules, and they do not have that center microtubule. And these 2 centrioles come together to form something that's referred to as the microtubule organizing complex, or MTOC, or organizing center, however you want to call it. And then we have these other ones, which are called the intermediate filaments. And these are what provide that tensile strength. So what helps keep that cell from being torn apart is the intermediate filaments. These have no structural polarity. And so examples of this would be the lamins, which are found in that nuclear envelope that we were talking about earlier. And we've got keratins, vimentins, and neurofilaments. And so, again, coming back to the nucleus, DNA is stored in genes that are transcribed to RNA, and this is happening in the nucleus. And we will say that's easy. The inside of the cell, made of water, enzymes, macromolecules, ions, dissolved gases, is called the cytoplasmic matrix. Within the nucleus, there are non-randomized distributions of DNA that are only visible during mitosis, and so these are called chromosomes, or chromosomal territory. These are the regions specifically. And then we have these things which are called our intermediate filaments. These are made of the subunits, and these are just long alpha helical protein structures that form dimers, and then these two dimers come together to form a tetramer, and these other tetramers will then join together to form even larger structures, and they bind laterally and then also end to end, and these form your actual intermediate filaments. Now we're looking at a picture of mitochondria. Mitochondria are what produces the ATP in the cell, the main energy source for the cell. And the key thing to make note of is that inside the matrix, so in the inside of this picture, so the actual, slightly lesser gray region here, that is where we're doing our pyruvate oxidation, the citric acid cycle, and beta oxidation of the fatty acids. So we've also got an additional thing here, which is blank occurring in the inner mitochondrial membrane. This is the electron transport chain. The electron transport chain is what's responsible for making that ATP, what drives that wheel, the ATP synthase, to actually pinch and force together that phosphate onto the ADP. And so the other interesting thing, or side note about mitochondria, is that they are called prokaryotic symbionts, and this means that they originally existed as separate bacteria, but these bacteria came together and now they live inside of their host. And the other interesting thing about mitochondria is that they do have their own DNA, and they do have some ribosomes that they're able to make their proteins from. However, they're only making about 1% of all that DNA, or of the proteins that they need. So 99% of the proteins that make up the mitochondria comes from its host cell. So mitochondria inside of a eukaryotic cell are using most of the eukaryotic cell's machinery and DNA to actually make their own proteins that they need to survive. And the nucleolus is where we're going to make our ribosomal RNA for ribosomes. And we've got the two types of chromatin, just reviewing this thing, where we've got the euchromatin, that is the loosely packed and easily reached DNA that can get readily accessed by RNA polymerase for transcription. And then we've got something called heterochromatin, which is that really densely packed stuff that is harder to express and it doesn't get expressed. And we coil the DNA around histones, we make it really tightly packed, and this is going to form chromatin. And so the other thing here that's happening is we're going to take mRNA to make proteins. So ribosomes are in charge of taking the mRNA, the messenger RNA, from the nucleus after it's been transcribed from the DNA. And ribosomes can live free in the cytoplasm, or they can live on the surface of the rough endoplasmic reticulum, and they're called bound ribosomes at that point. And then so the types of ribosomes that are responsible for making cytoplasmic proteins, nuclear proteins, and mitochondrial proteins are free, which means they just float around in the cytoplasm. They don't need to be bound onto the rough endoplasmic reticulum. And the other type of the ribosomes are called the rough endoplasmic reticulum bound ribosomes, and these are responsible for making the bigger proteins, the more complicated proteins. These are things that live or will be put into the membrane, those integral proteins or the peripheral proteins we talked about earlier. These can also be things like the proteins that are going to get secreted, so insulin would be one example of a protein that a liver cell would secrete, or we can also do the lysosomal proteins, and these are proteins that are going to be responsible for degrading other proteins. And so the other key thing here, coming back to that nuclear envelope, is that we know that it has many nuclear pores to allow for communication between the actual nucleus and what's inside of it and the outside of the cell. And we know that water and ions can move through easily, but the proteins and the RNA are things that are too big and they need to be actively transported by the cell. And we've got the double lipid bilayer membrane to make up this structure that we call the nuclear envelope, the barrier between the nucleus and the cytoplasm of the cell. And where we build stuff for vesicles, membranes, and exports is called the rough endoplasmic reticulum. And so basically what we have here are a bunch of these ribosomes that are bound onto the surface of this organelle. And we're also doing some initial tagging with sugars, so we call that glycosylation. And this allows us to actually know and identify these proteins as they're being manufactured, to know what we're going to do with them at the end of the day. So the rough endoplasmic reticulum is the important one to remember. And we have another structure or organelle inside of our cells that is called the smooth endoplasmic reticulum. This is responsible for lipid synthesis, making cholesterol and steroids, detoxifying things, producing gastric hydrochloric acid, and also sequestering, which just means storing the calcium ions. And an example of this would be in the muscle cells that do a lot of that contractions, so they need to have a reservoir of calcium ions for the cross bridges that happen in order to actually allow you to move your muscles. And this soluble monomer subunit of actin is called a G-actin. And the three types of cytoskeleton filaments are that actin, microtubules, and intermediate filaments. And we get these G-actin monomers coming together, and they form double-stranded helical fragments that cross-link to form these protofilaments. Actually not protofilaments, but this is just your F-actin. And then what happens is, on this next slide, we have proteins that get packaged and then further modified in the what's called collectively the Golgi apparatus. And so as the rough endoplasmic reticulum bound ribosomes are assembling these proteins, they then give them through these vesicles to the Golgi apparatus. The Golgi apparatus has multiple faces to it. It has a cis side or face to it called which is on the same side as that endoplasmic reticulum. And then we've got the trans Golgi side, or the trans Golgi network, and this is where you've basically got this shipping center. It's like an Amazon warehouse with a bunch of stuff leaving to go off somewhere and ship these proteins out that are being made by your RER. And then organelle movement, chromosome movement, and flagella or cilia structure comes from microtubules. And proteins attached to microtubules to move to the negative or positive end of these microfilaments. Dynin is what is moving towards the negative end, which is to say dynin takes you back to the center of the cell, and then kinesin is what takes you out to the outsides of the cell. Microtubules have these two subunits, which is called alpha and beta tubulin, that are forming their heterodimers that become those protofilaments to become cylindrical structures called microtubules. And when we look at vesicles that transport and store proteins that go from the trans-Golgi network to the membrane are called secretory, like you're secreting them, vesicles. And then we have an additional type of vesicle that is used for endocytosis. So as cells take things in, they form these things called endosomes. And we also have other types of vesicles called lysosomes and peroxisomes. And so another thing that we're going to come back to here is that membranes are things called amphiphatic, meaning they've got that polar head, nonpolar tail, so they've got those two characteristics. And we've got the integral proteins that form that entire, or they span the entire membrane, and then we've got the peripheral proteins that are just on the surface. We've got our double lipid bilayer membrane making up the nuclear envelope, and when you phosphorylate these lamins, what will happen is that they're going to disintegrate to allow things like mitosis to happen. And we've got three types of cytoskeleton filaments: actin, microtubules, and then we've also got the intermediate filaments. And these G-actin monomers are going to come together to form their trimers. Double-stranded helical filaments are going to cross-link to form F-actin that has a polarity, which means it's got these chemically different ends to it. proteins attached to microtubules to move to the outside or inside of the cell, and we know that kinesin is what moves to that plus side of the cell. And these microtubules, when we look at their structure, we're going to recognize that there are these things called protofilaments that will come together to form the entire microtubule, or sorry, the heterodimers. And heterodimers are what come together to actually form those protofilaments. And then we've got cilia and flagella. It's got that 9x2 pattern, and that was that thing called an axoneme. And when we look at centrioles, they are going to be triplets of these microtubules, but they don't have those central microtubules. And centrioles are cool because you'll get two of them coming together and they form the microtubule organizing centers. The rough endoplasmic reticulum is where we're going to build stuff for vesicles, membranes, and exports. Proteins are going to go through the pores and get tagged with sugars, which we call glycosylation. And then proteins are packaged and further modified by the Golgi apparatus. The cis side is that side that's on the same side as the rough endoplasmic reticulum, and then the trans side is where we're going to be moving stuff onto the outside. And then our cilia and flagella have 9 pairs of microtubules with 2 central microtubules. And so we've got these A and B tubules that form that pair, and these tubules are differentiated by the fact that they have different numbers of what is referred to as the protofilaments. Vesicles, there's different types of them. We've got the lysosomes that will break down the endocytosed macromolecules, and then we've also got the peroxisomes that are going to use hydrogen peroxide to digest fatty acids. And getting finally here, we've got the 3 types of cytoskeleton filaments: the actin, the intermediate filaments, and then also the microtubules. And we've got these centrioles. Centrioles are going to come together to form the MTOC, the microtubule organizing center. And then we are going to have the intermediate filaments. These are things that provide that tensile strength for the cell. They don't have any structural polarity. Examples are the lamins that we talked about earlier. And then we've got the actual microfilament structure here, talking about the subunits, how they come together to form the alpha helical proteins that are dimers, antiparallel tetramers, and then ultimately they become intermediate filaments. And finally, we're almost there, blank occurs in the inner mitomembrane. So we are going to have the electron transport chain happening in that inner mitomembrane itself, and that's where we're going to actually produce all that ATP. And then your centrioles have 9 triplets of microtubules without that central microtubule, and that is going to wrap things up. Thank you all for watching, and I hope this video was helpful. Feel free to use any of these resources, and I'll talk to you guys next time.